And so to kind of mitigate all these problems, we have an integrated solution, which is the G.Screen platform, with our 4 assets that we put together in order to be able to answer and resolve all these problems I talked about, especially when we are working with membrane proteins in antibody discovery. So the first step would be to use our G-mixes. The G-mixes actually are proprietary mixes. We have more than 220 mixes that allows us not only to extract the protein from the membrane, but also to stabilize them in order to be able to work with them in vitro. But we go a step further, actually, and we have developed an assay called the G.Select assay based on fluorescence that actually can tell us in which format the target is in a specific mix. So we can know, for example, for GPCR, if it's in an active state or inactive state, or we can know as well if it's in a dimeric form or monomeric form. We also went a step further, and we have made a library, and we did studies in lipidomics, where we actually extracted the lipids from 30 different cell types. And we know now we have the composition of all these membranes from specific cell types, and we are able actually to reconstitute the specific lipidic composition in vitro and re-embed the receptor in it. And this is actually very important for the stabilization of the target and in the specific conformation and specific environment to increase the selectivity and decrease the off-tissue responses. And finally, we had put together a functional assay, a cell-free, fluorescent-based functional assay that allows us actually to validate that what we have here, purified, reconstituted, is functional using benchmark ligands, of course, whenever they exist, but also to do some target validation, target engagement, and most of all, being able to characterize the candidates and the hits, the binders that we get. I wanted just to focus a bit about our G-validation assay and how it does work, because in the case studies, you will need this information to understand a bit more how do we work with it. So basically, this assay called the G-validation assay, it can be used for target validation, for target engagement, for profiling, and for potency assay. But it doesn't detect only binding, it also detects structural changes due to ligand binding. It's based on fluorescence, and basically what we can do is we have developed this small fluorophore that you can see here in yellow, that we can technically clip it anywhere on a specific protein. We, of course, always choose somewhere where we can detect an important structural change for the activation of a certain target. So here, for example, I represented a transporter where we have put it on the extracellular part, and we can then monitor the opening and closing of this transporter. You can also put it on the interaction interface between 2 proteins that can dimerize and detect dimerization or splitting of the complex. You can as well use it on a G protein coupled receptor. And in this case, for GPCR, we know that they all share in common this feature that the TM6 opens and closes when the receptor gets activated or inactivated. And actually, if we go to this specific example, how does it work? We have this fluorophore that has a very nice characteristic, is that its fluorescence changes in function of the environment in which it is. So if you first take a first emission spectra of this fluorophore, once you have added it to the GPCR, you will have the basal activation state of the protein. And if you add, for example, an agonist or a J-mix that stabilize the active state, you will have opening of TM6, and the fluorophore will be surrounded with more polar environment due to the buffer, and you have a shift in the emission spectra, towards the yellow here spectra. And if you add an antagonist or an inverse agonist, you inactivate the protein, and so the fluorophore would be buried in a hydrophobic cavity. Its emission spectra changes, and it go to this blue curve here. And so what is really interesting is that we can measure the shift in lambda max between the basal state and when we added a J-mix or a ligand and do, for example, dose response or kinetic studies and to compare candidate between them and compare them to the benchmark, calculate their IC50, KD, and Bmax. So what we provide is the target that can be either in our J mixes in a bicell form or in a nanodisc with a lipidic bilayer that are stabilized in a specific state, oligomeric or monomeric in a specific conformation and in a specific environment. And those guys can be used by our clients and partners as antigen for immunization, for selections, for screening, and biophysical and functional studies.